Anyway, today I will discuss two things. One is post-transcriptional modification, post of RNA, and briefly about genetic code. And this class will be of little shorter duration than the normal class, because there is not much to be talked about those two topics. I can tell, but all those things will not be required. It will be difficult to memorize. So in short, what you require, that I will try to tell. Basically, yesterday I told you that eukaryotic genes, particularly human genes, are very complex, and this RNA, which is formed from the DNA, these are of multiple types. Only three main types are mRNA, rRNA and tRNA. But there are many other types, as I told you yesterday. So all these need to be processed, because all RNAs are required. So the processing is quite different for all those things and way of functioning. But by and large, what are the post-transcriptional modification? That will include only four main points, because it's a very large structure which is formed from the DNA, that is called a primary transcript, and these primary transcripts contains both exons and introns. So these introns are to be removed, because introns are the non-coding part of the gene, and that RNA which is produced needs to be made stable. So there must be a mechanism to protect the RNA from being degraded. So that is the reason why post-translational modifications are done. And you see, the RNA is produced in the nucleus, but it functions basically in the cytoplasm. So it has to come out from the nucleus and function where it ought to function, as rRNA, as tRNA, or as mRNA. So there must be direction of movement of those RNAs which are formed from DNA. So this processing essentially helps in not only protecting and making the RNA stable, but also allows its movement to the right place for making its optimal function. So basically it includes 5' 7-methyl guanosine capping, poly-A tailing, third one is splicing, and fourth one is RNA editing. Normally we know that this DNA which is there, against which RNA is formed, and that particular information is not changed. But in RNA editing, this is edited at the mRNA level, even after replication, that you must remember. So this can be changed at that stage. So an altered amino acid can be produced, or a nonsense codon can be created, or at the splicing junction, proper nonsense or missense mutation may not be there. So what will basically result, end result? So from the basically their DNA molecule which is having both exon and interfering part is basically intron. These are non-coding part, and this is called also junk element. So when a transcript is formed, basically it forms the entire thing. So if a protein is formed from this structure, so that will include that coding sequence of the intron. So that must be removed. So one step is removal, first cutting the junction between the two and then removal and then joining together. That is what is called splicing. So this is also removed. This is called splicing. This is out. So basically you get exon and exon only. So this part is called splicing. Splicing basically means joining. But until and unless you cut it, it cannot be made. So how this is done, this cutting part? So it is done by mainly two parts. One is basically in the intron region. In the intron region, there is a consensus sequence that is UACAAAU, and there is one adenine. Basically, this is the branch point. This is 3' end, this is 5' end of this structure. Now, what it does, it makes a nucleophilic attack here, this adenine. So it makes a loop-like structure. So it cuts here and then makes a loop. And this point is about 20 to 40 base pairs ahead of the 3' end of the exon. So exon is cut here and it forms a lariat. Lariat means a noose, fast, Bangla fast. And then finally it is cut and the entire structure is removed. This is one way of doing it. So both the exons are joined together. So this part is totally removed. So two exons are connected to each other. That is one part. Second is use of spliceosomes. I will discuss it in this, you will see it in the slide that I will tell you. With the help of spliceosome, this also can be cut. That is another way. Then after doing these things, this part has been formed. So then this is 5' end, this is 3' end. So this part is subjected to exonuclease activity, degradation, both 5' exonuclease and 3' exonuclease. So to prevent this attack by the 5' exonuclease, 7-methyl guanosine capping is done. So what it does basically? So at 5' end you are having a nucleotide RNA tripeptide, triphosphate. So a 5' RNA, 5' RNA triphosphatase enzyme first cleaves and removes the diphosphate. So basically at the terminal part, the terminal nucleotide, this 5' RNA triphosphate is acting, and then since guanine is required, guanosin will be required, so guanosyl transferase. Guanosyl transferase enzyme will come here. It will attach guanosin moiety to this end. After that, since the 7-methyl is there, so 7-methyl transferase. So this enzyme will act. So all these 3 enzymes are called capping enzymes. The unique part of this capping enzyme is that it doesn't function optimally without RNA polymerase II. So with the when the RNA polymerase II is there, all the 3 enzymes will be active, and with the help of all 3 enzymes, capping enzymes, 7-methyl guanosin capping will be done. Is this part clear? So first cleavage by 5' RNA triphosphatase, and then guanosyl transferase, and then 7-methyl transferase, and all these 3 are called capping enzymes, and they will act when RNA polymerase II will bind to it. So once this is done, so this 7-methyl guanosin capping is done here. So this protects the RNA from 5' exonuclease activity, number 1. Number 2, it makes it stable, because somehow it is stable, it is prevented from degradation. Third one, it has to come out from the nucleus into the cytoplasm, to the ribosome. So it will help in that process. In a similar way, at the 3' end, poly A tail will be added. Poly A tail. Poly A means adenine, adenylic acid. So basically here adenylate polymerase enzyme will be there, and this will cause a poly A tail. A. This is called, what is poly A tail? So again, this poly A tail will prevent it from being degraded by the 3' exonuclease activity. Now the function of this poly A tail is to direct again to come out from the ribosome and settle onto 40S ribosomal subunit. You see, humans are having 2 subunits, one is 40S and one is 60S. So it locates at 40S ribosome. So one is splicing, one is 5' capping, one is 3' polyadenylation, and fourth one, at times the methylation also takes place at adenosine moiety, methylation. Methylation can take place with adenine in some cases. And fourth point, that is RNA editing. As I told you in the beginning that normally the information which is transmitted from the codon onto the mRNA or onto the is not changed. But here it is changed. Why? You see, one good example is ApoB gene. One good example, ApoB gene, you know one thing is ApoB 48, that is 48, ApoB 48 and ApoB 100. Means 100 kilodalton, molecular weight is 100 kilodalton, 100 kDa. And this is 48 kDa. That means it is almost 48% of that one. So this is a truncated version of the protein. That means it is if it is the whole protein, it is a truncated version. And how it is done? It is basically, this is having a nucleotide base pair of about 14,500 base pair. So at 6666 position, basically there is change of one codon to a stop codon through mutation, that is cytidine, change to uracil. Through cytidine deaminase. Cytidine deaminase. What it does basically, cytidine converts cytidine to uridine. So U is formed. So now you see it is the nucleotide is changed from cytidine to uridine and it makes a stop codon. So the synthesis will stop somewhere in the middle, nearly 48%. So that will generate 48, ApoB 48. And why it is done? It is done deliberately by the nature, because ApoB 48 is expressed in the intestine. It is a part of chylomicron. ApoB 100 is formed in the liver, which is the apoenzyme for LDL. So this is for chylomicron. This is for LDL. Same thing can occur in multiple proteins, because you see one protein can exist in different forms and in different tissues they are expressed at different times. So that can also be accomplished by RNA editing. So this is what is about. I can tell you, is it a slide? Just you can see the slide, you can now note it down. See, post-transcriptional conjugation. See, basically it is a large structure. After the primary transcript, it is about 10 to 50 fold. So that structure needs to be shortened somehow. So that is done by basically endonuclease cleavage. Endonuclease means, yesterday I told you, anything, any sequence, in the entire sequence, any enzyme which is cleaving except two ends is called an endonuclease. And extreme part is exonuclease. So during processing, endonuclease activity must be there. And then poly A tailing I have told. 5' capping I have discussed. Methylation or N6 residue and the two hydroxyl group of ribose, they get methylated. Removal of intron I have told. Splicing of exons means joining together I have told, and RNA editing. These are the basic points for the quarter. Now you see that what I have told you, now this is in short. This is what I schematically exons and introns. Now primary transcript, now adding of 5' capping, polyadenylation, RNA splicing and joining. You see the mature RNA with 5' capping. So now one part I did not tell during this time, that snRNA, the spliceosome part. Yesterday I told you about there are only a few of the snRNAs, important snRNAs. You see some are present in the nucleoplasm, some are in the nucleus, some are in the chromatin. So basically, out of all these, only 5 uracil rich snRNAs, that is U1, U2, U4, U5 and U6. They are in association with more than 60 small ribonucleoprotein. They form spliceosome. In that structure, what you have seen, that in the layered structure, that same things sequence will be there. So here what happens, this 5 U1, U2, U3 is not there, U4, U5 and U6. See that sequence UACA, this is about 20 to 30 base pair ahead. This sequence UACA, AAC, this part, this adenine, basically this E1 binds to this part. U2 binds here. Now it forms a structure, it clips here. So U1 and U2, it forms a layer again, something like this, to which U4, U5 and U6 join. U4, U5 and U6 join. Finally, U4 is removed to make it functional, and U1 is replaced by U6. So, first U1, U2, U4 and U5, that is an unstable complex. After that, this U4 is clipped. Then in place of U1, you get U6. So now the structure is basically U2, U4, U5 and U6. This structure causes removal of this from this part. That is what is called spliceosome. What structure is that? Now you see here. You see this intron is there. So snRNPs I have told you, small nuclear protein, ribonuclear proteins, there are more than 60 proteins are involved. So some proteins will be involved here, and they are excised. It is shown schematically here, but the entire process involves the 5 snRNAs and a good number of, that is more than 60 snRNPs. So this is what is a spliceosome. This is what is a spliceosome, and by this mechanism, so there are two mechanisms, one is late formation, the other one is, there is a spontaneous intrinsic one, it does not require spliceosome. This is spliceosome mediated, that is cell mediated. This is the two differences. What are the? So alternate splicing, as I told you, so splicing, if there is a problem, suppose there are 3 exons, 1, 2 and 3. Now normally the codon sequence must be very much appropriate here, otherwise cutting will not be done. If there is a mutation, then of course it will not be cut. Cutting will occur at the nearest, second nearest position. So it can happen between that 1 and 2. If it is a normal 1, 2, 3. But if it is not so, then it can be 1, 2, 3. It can be between 2 to 3. If there is a problem here, it can be 1 to 2 to 3 and 1 to 2 also. Based on the mutation here, so combination can occur in various proportions. So that will cause an altered protein synthesis, and that will be a non-functional protein. So one cause is beta thalassemia. Thalassemia, of course, there is a very big book of about nearly 4 kg on thalassemia syndrome. That's a big book. I tried to digest, but after seeing the book, I closed my eyes simply. And again, expression of glucokinase gene. Glucokinase gene is present in the pancreas. It is basically producing insulin, so it must act as a sensor. So glucokinase enzyme is there, but there is a differential promoter site of glucokinase both in pancreas and in liver. Liver is expressing hexokinase, liver is expressing also glucokinase. So there is a differential promoter site, as I told you yesterday. So the promoter site also, first and foremost thing of any transcription to occur, promoter site, everything must be completed at the promoter site. So if there is a differential promoter site, then there will be a defined expression, and that's what happens with glucokinase gene also. What are the? So RNA editing, this part I have told you. So what happens here? The premature stop codon. This you note down, the cytidine deaminase, catalyzed deamination to form cytidine to uridine. This I told you. So basically CCA to EOA, EOA is a stop codon. So that is the actual reason why it makes a truncated protein of 48. What is the reason? So to summarize, this is the basic summary. You must note down as a short note if it comes in the exam. So one is 5' 7-methyl guanosine capping, protecting from 5' exonuclease activity, making it stable and allowing it to move to nucleus to cytoplasm. Polyadenylation again prevents from exonuclease activity at 3' end, and it is allowing it to come to the 40th ribosome of mRNA. Splicing means removal of joining. RNA editing I have told. After complete processing, then only it comes out. You can add methylation also if you want to include in that part. So basically these are the 4 main transcriptional, post-transcriptional processing. After this thing, the RNA functions properly. Of course, there are post-transcriptional processing for microRNA, snRNAs, tRNAs, rRNAs. So they will differ according to their function. Now, briefly I will tell you about genetic code. All of you know it because this was taught at class 12 level. Just a few things you have to remember. Perhaps all these things were not told in your class 2 level. So, what is genetic code? What is the difference between the genetic code and genetic codon? Actually, the genetic information is stored in the DNA molecule. The entire information, that is what is called genetic code. And DNA is the director. DNA directs everything. So, codon is basically a 3-letter combination, which basically codes for a particular amino acid or a stop codon. That is what is a codon. So, there are 4 bases, as you know. So, again I am telling you, this coding system has been done as per sequence of mRNA, not as per sequence of DNA. So, that is the reason U is present in part of codon, not T. Remember, I repeat, and the codon table is followed, which is an RNA codon table, not a DNA codon table. So, there are 64 different codons which are possible. So, we are having only 20 amino acids, and 21st amino acid is the selenocysteine. So, out of 64, there are only 3 stop codons. So, UAA, UAG and UGA and UAA. So, 61 codons are there and 20 amino acids. So, there must be overlapping. So, as I told you, codon is basically a triplet codon. Either it signals a stop codon or a specific amino acid. And standard codon table, I am telling you, standard genetic code is as per RNA codon table. And it is the mRNA which directs the protein synthesis. So, translation is again made by, as per standard genetic code, is translation table. There is a DNA codon table is there, but DNA codon table is not followed. What are the Regina? And now you look at this structure. So, this you will get it in the book everywhere. Only some part I have written deliberately to indicate you. See, only there is one initiating codon, that is AUG. 64 codons, 61 basically codons are there. 63 stop codons, remove it, 61. So, there are single codons are there only for AUG and UGG. One is for methionine, one is for tryptophan. But there are double codons for at least, you see, phenylalanine is double codons. You see, this is what I am trying to indicate: phenylalanine, histidine, glutamine, asparagine, lysine, then glutamic acid. They are having double codons. Triple codon, that isolation, you see 6 codons are there. 6. Triple there is 6 codons are there. 6 codons আছে, লিউসিনে আছে দেখো, আর্জিনিনে আছে, সেরিন 4টে এখানে, 2টো এখানে. So, they are having 6 codons. 4 codons are there for valine. That is valine is 4 codon, threonine is 4 codon, proline is 4 codon. 6 codons are there, there are 3 stop codons. So, basically, if there is more than one codon, that is what is called degeneracy. Normally, one codon represents one amino acid, but here you see the same amino acid can be coded by multiple codons. One codon codes for one amino acid, but multiple codons can code for the same protein, same amino acid. That is what is called degeneracy. Multiple things are there, excess. That's why it is called degenerate. More than one codon for one amino acid is called degeneration. Now, these 6 points you have to remember. These unique features of genetic code. One is triplet. That means it is a 3-letter coded combination, that is universal. That is true. Say UG, GUA, CCA, whatever way you say. So there are successive 3 nucleotides, and again I am telling you this is as per codon table on the mRNA. Ageta dekho to, the table dekho. See, look at this structure. U is here. If it would have been as per DNA codon table, then instead of U, T would have been there. So this indicates this is as per sequence of the mRNA. And during protein synthesis, I will tell you again, and I will show it to you why it is important. But it has to match. So it is universal, means this codes for bacteria, this can code for yeast, this can code for mammals, this can code all living organisms, plant, even human beings. So that is universal, that is the same codon is coding the same amino acid in all species. But you see, everything is not that absolutely true. So there are some exceptions. And mitochondrial genes, there are exceptions. In a short while you will see. In mitochondrial genes, mitochondria is having a separate machinery altogether, having its own DNA, own RNA and own protein synthetic mechanism. That I will tell you during translation part. So this part, that universal nature, has been preserved during evolution. But as our knowledge expands, we understand it better. Because when it was discovered, perhaps this mRNA part was not known. It was also a fact that mitochondrial DNA discovery was known much later than when it was originally made. Now, degenerate, as I told you, it is representing more than one codon. And fourth point is non-ambiguous, means one codon codes for one amino acid. There is no ambiguity. There is no ambiguity, that is called non-ambiguous. And then non-overlapping means codons, when it is read from 5' end to 3' end, there must not be overlapping. Once you read this 3 letter, you cannot read that same letter. So there cannot be overlapping. So that means adjacent codons do not overlap. As I told you, that must be read here. You cannot read this part, and then this part. No. This has to be 3 letters from one end. And this is represented, that is what is called non-overlapping. That means this reading has to be in a continuous manner. And non-punctuated means this is comma-less. There is no comma here. You can see there is no comma. So there is no punctuation. That is the reason why it is called non-punctuated. So it is easy to remember. See, non-ambiguous part I have written deliberately at the 3rd, 4th and 6th position. So first you remember triplet, universal, degenerate, non-ambiguous, non-overlapping, non-punctuated. That will be easier to remember. This 6th point you must tell at a stretch. What is রেজিনা বলে আমি দেখতে পাচ্ছি না মোবাইল ছাড়া কেউই বাঁচতে পারবে, মোবাইল চলে গেলে আমার মনে হয় জীবনটাই চলে যায়, তার চেয়ে ভালো। ঠিক আছে, এই আদার ফিচার্স বলতে বলছি যেটা পোলারিটি, what I told that you have to read from one direction, that is 5' to 3' direction. There is one start codon, you remember. It can come as an MCQ, AUG, stop codon, UGL, UGA and UAA. These are the 3 things. In many books, these are written as amber, ochre and opal. They tried to find out why they have been named so. It was not during our time. I found that this UAG codon as a stop codon was discovered by Charles Steinberg and Richard Epstein, and they named it peculiarly after the name of the train, and it was the German meaning of that name of his train. So that is the reason it is called amber, and UA called ochre because it is the same color thing they have made, and UGA opal, again the same color thing, mineral color they have made. I mean, there is no need for remembering all those things, but I have seen some books are also mentioning it. That's why I tried to find out what is the actual reason behind naming these top codons as amber, ochre and opal. So mitochondrial DNA, you see here one important point. In mitochondria, there are tRNAs, there are mitochondrial 3 and cytoplasmic 31 tRNA is present. Mitochondria is having altogether different genetic composition and genetic RNA, DNA, protein synthesis. So RNA, mitochondrial RNA is 22, cytoplasmic RNA is 31. See here, look at this here. See, normally you see UGA is a termination codon. UGA is a termination codon in cytoplasm, but in mitochondria it is coding tryptophan. AGA and AAG, it is a normal cytoplasmic, it is coding for arginine and arginine, but both are acting as a termination. AUA is a leucine isoleucine that is coding for methionine. So what we told that it is universal, that genetic code is universal is not true as it is proved in the mitochondrial DNA itself. So cytoplasmic stop codons are UA, UGA and UGA, and you see here the termination codon is AG and AGG. So it is not matching. What is So Ubel, just note down one point, Ubeling or Ubel phenomena. See, there are 6 codons for leucine. There are 6 codons for leucine. Suppose here it is a sequence of CUC. Now, normal pairing C should combine with G, but here it is combining with it is U here, G is here, CEU. So basically it indicates that freedom is allowed at a third position, means somewhat we are having a little bit of lackadaisical attitude about the third codon. That is what is called Ubel phenomena. Freedom is allowed at the third position, and 4 main Ubel base pair. Remember these 4 main Ubel base pair. One is guanosine, guanine and uracil. One is hypoxanthine and uracil, hypoxanthine and adenine, and hypoxanthine and cytosine. These are the 4 Ubel base pairs. What are they? So 64 codons, 45 tRNAs, 3 stop codons. Freedom at third position is there deliberately, because to adjust all these codons, because if there is slight mutation, that can be managed if there is available codon, same codon for that, same amino acid is available for the same codon, which has been altered even by mutation. Some exceptions, as I told you, AUG is normally initiator codon. It is perhaps last slide. In a few, GUG is the initiator codon. AUG in eukaryotes it codes for methionine, but AUG in prokaryotes codes for formyl methionine. And mitochondrial DNA exceptions you note down. So UG is a terminator codon in nuclear DNA, but arginine and mitochondrial DNA. And that is all about DNA part of the army. So these 6 points of codons, that is, you must remember, and you must know the Ubel phenomena also, that there is freedom is allowed at the third position, and this can be actually managed without causing any alteration of the amino acid sequence because of degeneracy of the codon. Multiple codons are available for the same amino acids. So by chance, if it happens to in that position the same type of mutation, nothing will happen. But if there is a problem at a particular amino acid like sickle cell anemia, then it will definitely cause disease. Okay? So I think it is okay. If you have any question, you can ask. If you have any question, you can ask freely, which I will be able to tell. Again, I am telling you, I am happy, I will be happy. I want one question, ask a difficult question to me for my own reading. Otherwise, I will never learn. Ask a question or anything you could not understand. Okay, then let us make an end.